Good evening. It is the 12th of March, 2025, and this is our third journal club, second student journal club overall for Holy Cross High School. And today we have Lila Ashton, who is a senior class of 2025 from Morris Catholic, who will be leading the discussion. Lila Ashton is presenting Sex Biasing Influence of Autism Associated, UBE3A. How are we pronouncing that, Colin?
Ubethria.
UB3.
UBE3A, please.
No, I love that, the eubethria. Gene overdosage at connectomic, behavioral, and transcriptomic levels. This is from the Gozzi lab, which is in Italy, Genova. And it's published in Science Advances, which is the answer to Nature. Nature had a similar thing where they would come out with papers ahead of time, but this became its own journal. Lila, the floor is yours.
Sure. Did David want to present? Because I don't see the presentation, but I might just not be able to see it.
Yeah, I'll start there.
Okay. So just go to the second slide, David. David. Okay. So just really quickly, Dr. Char wanted me to put in a picture of the autism triad, just in case people are unfamiliar with the common symptoms of autism. So I think the picture itself is a pretty good overview, but basically there's three different things in the triad. So it's social interaction, communication, and it says here activities and interests, but sometimes it's also called the restricted and repetitive behaviors. So social interaction is stuff like, you have trouble, making and keeping friends. Also, when you're younger, you can tell like if they struggle with teamwork and stuff like that. They often have difficulty understanding social cues, like body language or facial expressions. Often people have a very flat face when they're speaking. For communication, that's kind of similar, where like they have trouble understanding sarcasm or jokes or indirect language kind of immediately. So they may interpret things very literally until it's explained to them. And they might have delayed speech or unusual speech patterns. And the last one, I kind of mentioned this before, but the picture I put says activities and interests, but I prefer the restricted repetitive behaviors because that includes, kind of like stimming, like rocking and hand flapping, kind of repeating actions. But it also refers to like how they often, people with autism typically dislike change and prefer strong routines. But there is that idea of like an intense focus on certain interests or topics. Also, before I might have to go back a second and kind of delve into the title to give a preface, but Dr. Sharp mentioned he might want to go over the Engelmann syndrome and Prader-Willi syndrome dynamic. I don't know if you would want to do that now before I get into the paper or not, but
No, I think you want to introduce your the title like you wanted to.
okay. David, could you go back to the title slide really quickly? So I actually really like this title, kind of because it feels a little bit like a three-pronged thesis in like English a little bit, but it's title. So there's this gene called, are we saying UB3A? Okay, I would say UB3A. So it codes for a protein that goes by the same name. So you might see those used almost interchangeably. But that protein is a ligase, and you might already know this from bio, but that's kind of an enzyme that sort of regulates which proteins get broken down and kind of ensures that extra proteins or faulty proteins don't start to like accumulate in neurons. So if either the gene or the protein isn't working properly, neurons can kind of get overloaded with too many or too few proteins. So going back to the title, what you're going to learn is that UB3A overdosage is going to do different things to males and females. So that kind of takes care of the first part, which is the sex biasing influence of autism-associated UB3A gene overdosage, because you understand what UB3A is, what kind of generally happens with overdosage and the sex biasing part. And the part where it says connectomic, behavioral, and transcriptomic levels part is what we're going to cover in our figures. So connectomic is going to be figures 1 and 2, which is David and Tyler, I think, maybe Cameron a little bit. Behavioral is figure 3 with my queen Maggie, and transcriptomic is going to be figures 4 and 5 with Colin and Cameron. And the experiment's going to be explained in detail by everyone else, but they had male and female mice, which it was both wild type, which is their control, mice, and the experimental group, which is the group of mice with the overdosage. So we can go into slide 1 now, but right before we do that, I might just want, yeah, slide for figure one. Sorry. Um,
No, wait.
hmm,
Before you do that, now if I can just provide a little
perfect.
background. Um, this is for those of you who are going to eventually, and if you're staying in the Science Academy, you're going to do this. Take my ECE course. We talk a lot about this because my area of research, my like subfield, very basic subfield that I was really interested in was how the environment affects or interacts with the genome to cause kind of make you who you are. And if you read any of my stuff, you can kind of find that is a pervasive theme. This is a very almost outrageous example of how unbelievable that effect is. So there is you guys, all you freshmen have had this already, right? The four macromolecules, the major classes of biochemical molecule are carbohydrates, lipids, proteins, and nucleic acids. And proteins are made of amino acids, but they are coded. The recipe for them is found in your genome as a gene. So a gene is a recipe for a protein. So UBE3A is a gene that has the recipe for a type of protein called a ubiquitin. And the ubiquitin takes out the trash. Imagine that you're constantly making proteins and you're making proteins and they do stuff and you're making proteins and they do stuff and you're making proteins. At some point, your whole cell is going to fill with proteins and nothing's going to be able to work. So you need a system that breaks them down. Ubiquitin is the trash can that does that. Okay. So, so there is this weird, and I'm going to say it's weird because 1 to 2% of autism cases, in a very rare type of autism called Angelman syndrome, that is caused by a mutation in this gene, but only if it's from your mother. And this should be mind blowing to you, okay? If you inherit this mutation from mom, you get this disorder called Angelman, which is characterized by significant intellectual disability. Okay, IQ is probably below 50, and stereotypic movement behaviors, stereotypic um, facial features, and it's a really debilitating disorder. And that's if it's from mom, and you tend to also find it in older mothers. So as a woman ages, her eggs are more susceptible to having mutations in any gene, but this particular one for some reason. And it's probably because if you have a mutation in this gene, you can still live, whereas mutations in a lot of other genes might kill you. Make sense? Yeah. Okay. So let me just say, because I finished this explanation, but David, go ahead and unshare the screen for a moment. Good. Okay. So if you get that same exact mutation from your father, you end up with a completely different disorder. So I want to show you. Let me see if I can actually do this for you. Can you hear the audio?
I
Can you
don't
guys hear
think
the audio?
so. Unless
No?
yeah, I don't think so.
Well, you don't really need the audio. Forget the audio. I'll just mute it. I just want you to look and what you're going to see is stereotypic facial features. Look at their hands, right? Facial features. They all have the same facial features. That's just not because they're related. That's actually what you'd find in across anybody. So that's Angelman, significant intellectual disability. It's pretty brutal. But the other one is called Prader-Willi, and that's from if you get it from dad. And let me just go ahead and find. So this guy right here has Prader-Willi syndrome. So these people, 100% of these people are morbidly obese. Okay, they're morbidly obese. So they start as babies. They're super floppy. Like if you try to pick them up, they're like, and they have no muscle tone in their necks or backs or anything. And they really struggle to thrive. And they don't really eat a lot. They're really struggling. And then all of a sudden something switches on and they become just inconsolably, irrevocably starving, and they want food at all times. They are so hungry all the time. They put on a lot of weight really quickly. When they get older, they're so desperate for food, they will steal food from stores, like, and hide it from their parents. And if you go into the room of somebody with Prader-Willi, you will find food in every nook and cranny because they're always hungry. And this is the same gene. Intellectually, they are different than a full-on intellectual disability. Theirs is not as severe as Angelman syndrome. They are probably an average IQ in the 70s, maybe up to 80, 85 at the most. And then, so it's not as severe as Angelman, but it's slowed down relative to their peers. Um, and that's just because of who it came from. Did it come from dad or did it come from mom? And that should blow your mind. Like, why would one mutation have different effects depending on where it came from? And that has to do with the fact that genes aren't alone. They interact with the world, they interact with the environment. So now we can go to figure one. And you take
Before
over, David.
I was gonna say before David starts, could I just say something really quick about hyperconnectivity and stuff like that before he gets into it? I'm assuming that's a yes. Since David's going to talk about hyperconnectivity and reduced connectivity, both like in males versus females, just really quickly. Um, so just to explain that a little bit, we're born with like way too many synapses. So synaptic pruning is basically this process of removing weak or unnecessary connections to make brain networks kind of more efficient and specialized. So when David talks about hyperconnectivity, I kind of like to think of it as like teenagers having way too many side quests when it comes to like making a certain conclusion. Their brains are kind of more creative, but they're a little bit less efficient because their synapses aren't pruned yet. So when you learn about reduced connectivity, I just assume you may have trouble coming to certain conclusions at all. But yeah, David's going to talk about the difference between those two things. But I just wanted to bring that up really quickly.
Okay. Can everyone see my screen?
Not yet. Oh, there it is.
Yeah. All right. Okay. So, okay. So we're gonna start off with figure one. Um, and it's basically just a bunch of different graphs comparing connectivity in different regions of the brain regarding males and females. So starting with part A, this is just a average result between males and females. No distinguish. They were not distinguished, so it's just a combination of the two. The darker blue regions represent regions of higher connectivity, or sorry, of lower connectivity, or also, as we said, hypoconnectivity. Um, and it's visible in regions of the thalamus and hypothalamus. So as you can see, the connectivity in the hypothalamus was a bit lower. Doesn't seem too significant, but a bit lower in the hypothalamus compared to the thalamus. Um, and taking a look at figure at part B, um, we have, oh, wait, actually, sorry, let me jump back into A. The comparison you have between the wild type and the UB3 gene, you can see that the UB3A gene does appear to have lower connectivity in both regions, both the hypothalamus and the thalamus.
Can you tell us
And
what those do?
the hypothalamus, or you're talking about both regions, right?
Yeah.
So the hypothalamus, I believe is mostly involved with like hormone regulation, and the endocrine system. I think also plays a role in the circadian rhythm. I'm correct?
I've heard that. Yeah, I've heard that. Yeah. Homeostasis in general, sure.
Yeah. Whereas the thalamus, I think, is more of that just kind of it controls, I know, some of the sleep wake cycle, just sensory processing, um, roughly. I don't know if you want to touch on that a little bit more.
Yeah, I'll say it a different way. So the thalamus is kind of the railway station through which everything flows. So if you have information coming from your body up into your brain, it has to go through your thalamus. If you have information going from your brain that's been processed and has to go out to your body, it goes through your thalamus. Even between regions of the brain, it goes through the thalamus. So that's kind of the railway station where it sends everything. And so, and talking about like what is it? What does it mean to have autism? It's really hard to describe it to people who don't have autism. And I think a really good analogy, hopefully you guys will understand what an analogy is, but a really good analogy is like imagine that the imagine that the prefrontal cortex is the conductor of an orchestra and it needs to have control over which instruments, the other parts of the brain, are actually active at any one time, what we're actually paying attention to. And people with autism don't have that control, possibly because of what's going on in the thalamus. Essentially, the prefrontal cortex may be trying to control it, but the thalamus is so misconnected that it's not sending the signal, and so everything's on all at the same time. Imagine the most overwhelming place you've ever been, or something that's really overwhelmed you because it was overstimulating, and then multiply that by 10 to 100 times, and you have kind of what a person with autism has to go through. And so the thalamus is kind of helping, regulate that.
Um,
Good, Dave.
and yeah, so obviously we can see why that plays an issue when you have that lower connectivity that's gonna heighten those symptoms. Okay, so taking a look at part B now. This is where they started comparing the male and female mice. You also have your control group still here, the wild type, compared to the UBE3 A gene. And
It
so
might be easier to refer to the UBE3A2X, which is a duplication. It might be easier to just refer to them as mutants, right?
okay.
The mutant. But also, I think we need to clarify what fMRI is.
Yeah, I wasn't 100% sure on that. I tried like I wasn't completely sure.
Okay. Does anybody know what it is and want to take a shot at
I,
it?
I I control F it like in the thing imaging. It says, is it functional magnetic resonance imaging? Something like
Yes.
that. Yeah.
Yeah. Well, y'all know what an MRI is, right?
Yeah, I just wasn't sure what the F for it meant.
Yeah. So an MR, as we in the biz call it, an MR, magnetic resonance image, is using magnetism to be able to look deep inside of the body, and it has really, really good resolution, so you can see soft tissues and stuff. It's one of the most important advances in medicine ever. Um, but they have for for neuroscience, they have developed a method, and this is a while ago. This is probably 20 years ago. They developed maybe 30 years ago. They developed a method where you could actually trace the blood flow to different areas of the brain and overlay it on top of the MRI and then you'd be able to deduce what parts of the brain are being activated at that time because of the blood flow. So if I show you a picture in an MRI, in a functional MRI, and I happen to be looking at your visual cortex, the blood supply will increase to the visual cortex, showing that you've activated that. So that should make the rest of it make sense, right? So it's essentially it's correlative. So correlation is not causation, but whenever there seems to be an increase in blood flow somewhere, it seems like that thing is activated. Make sense? Okay.
Okay.
And they did this for a mouse, which is insane. Do you know how expensive those machines are? This was a very expensive study. Very expensive.
All right. Well, so continuing on to part B of figure one. This was, like I said, the comparison between the males and the females, now directly. So you can see the connectivity, I believe, was it the red and the yellow, I think was again hypoconnectivity, Lila, if that's correct.
Yeah, they're in the yellows because you can see that those are positive,
Yeah.
so that there's
Okay.
more connectivity. Yeah.
So yeah, as you can see, based on the graph here, the females, the wild type by far had the highest connectivity, whereas the females with the mutant gene had actually lower connectivity than the males in the wild type. Not significant, but I mean, still, there's a difference there between the wild type and the mutant gene, which
Why do
is
you say
kind of
that?
so.
Why do you say that? Why do you say there's a difference if it's not statistically significant?
Oh, I'm just saying, like looking on the graph, but obviously graphs can be misleading.
I was gonna say, I think the paper did say significant reduction in connectivity. I think I might have said something like that in there, but I think since they have the asterisks there, it's significant. But like I see what you're saying, like it doesn't seem like that big, like decimal
Well,
wise.
this is a bit of a problem I have with some of the future figures is notice that they're I don't like I don't like how they this is nitpicky, but I don't like how they did the interaction. So they're showing the main effects with the little brackets like you see over an A, and then they're showing another main effect or the interaction by showing the big bar at the top where it says hypo. And I like, what does that even mean? Does that are they saying males versus females? Are they saying wild type versus mutant? Or are they saying as a result of wild type and mutant, there is an interaction with males and females? And if you read it, it's an interaction, but that's not what this that's not what that bar shows. So it's kind of a that is not how I would have presented it, but what do I know? I teach at Holy Cross. That was a burn on you, Holy Cross people. Yeah. All right, go ahead.
Okay. So yeah, now moving on to part C and D. These were just individually looking at the female mouse brains and the male mouse brains. So uh part C, again, those darker blue regions show that hypoconnectivity um, in the thalamus and hypothalamus, as well as here we also have some visible in the midbrain as well. So, and once again, you can see the wild type had, for the females, had higher connectivity in both the hypothalamus and thalamus. This is also visible in the male's brain, except here it's taking place in the connectivity. They're showing it in different parts of the brain. So here they have the ventral diagonal band and the prefrontal cortex is showing that increased connectivity. So you can see obviously in the mutant gene, the males in both the ventral diagonal band and the prefrontal cortex have that higher connectivity, whereas in the wild type, it's lower.
Yeah, I don't know if anyone wants to like jump in on that, but yeah, I agree with David. I kind of summarized it by saying like that females had reduced connectivity because of like probably excessive synaptic pruning. And then males had hyperconnectivity, like David said. Just you can see based off of the bar just at the bottom, like the blue versus the orange, the positive versus the negative. Um, so the hyperconnectivity would mean like the certain pathways are extra strengthened, but I think that the hyperconnectivity often leads to symptoms that are more associated with autism. So I think we're going to get into that more in figure 3, but yeah, I just wanted to add.
Can I steal for a second?
Yeah, go
I want to
ahead.
show you guys. I want to show you guys something cool. Replace the current share. Boom. So this is a. Can you guys see it? Yeah. This is an image comparing a wild type neuron, a basic wild type neuron, and all of the little branches and synapses to the neuron from a mouse model of autism. Actually, this may not be mouse, this may be human. But I've seen this many, many times. Notice how many more connections there are. And this is what Lila was talking about in her intro, where people who have autism tend to have many, many, many more connections than what you call a neurotypical brain, right? Hopefully that kind of helps you visualize what's going on there. Now, make sure you all remind me to, at the end, to tell you what the silver lining is about puberty. Okay? That's important. That's an important take home. I don't, Lila, I think you know it, but you probably haven't made the connection yet. So, yeah. So I just wanted to show that because this is what we're talking about. This is presumably what you're seeing at the cellular level for these mice.
Cool, cool. And I think figure two, unless anyone wanted to add anything else for figure one, figure two is Tyler.
Yeah.
I had a question about figure one, actually.
Okay.
I think I actually had two questions, but one of the questions like doesn't make sense. Um, I'm confused about the usage of color in all of these heat maps.
Yep.
Maybe I'm just dumb, or maybe everyone else is
No.
confused about it too. But
Yeah.
what is the difference between the dark blue and the light blue, let's say in just A, for example?
No idea. I was confused too.
Okay,
I
Lila was flaming me because I was confused.
I
I
was
mean,
not
so
flaming you.
I don't totally understand it either, but maybe it's just pointing at the extremities.
Like D makes sense to me. Like in my mind, it seems like on one end, like maybe on when I'm looking at
correct.
D, the way I interpreted that was that the reddish region was the UB3A activation, and then wild type was more activated in the yellow regions. But I don't even know if that's correct.
No, I agree with
I
you saying.
would
Sorry, you go.
no, I'd rather you go. Go ahead.
No, I was gonna say, I feel like for particularly this figure, it was made easier if I just ignored certain things like a little
Mm,
bit more, especially like
hmm
for A and B, just like not looking at the graphs made my mind pure. But then also like, yeah, for C, just looking at
Were
-2 and -5 and not like the actual scans was easier for me.
the scans necessary in this paper?
I guess for
Was it
identifying,
necessary?
for
No.
identifying what spots, I mean, like it's more
You
precise, I
wouldn't
think. I don't.
you wouldn't you wouldn't believe me if I just told you in the paper that it's in the hypothalamus. You need to actually see a drawing of it.
I guess not, but I guess since some of them are like more spotted, it would be useful to get a little look-see. I don't know. I don't think it's like a must-have, but I feel like it's, I don't know.
I kind of felt like it was gratuitous. It was they added it because it looks cool. It looks cool to have those MRI images because those are not real. Those are the they have if you read this they use 20 animals per group, 20 wild type, 20 of the mutants. And so what you're looking at in those images, and it's an image, it's not a photograph, is the combination of all 40 of those brains. And then they overlay some color on it. And I agree with Cam. D makes sense to me. But then if you read what is that color strip for B, it says that it's showing the interaction.
Yeah,
So
it
then I
just
started
doesn't
to
make any sense to me. I don't get it at
I,
all.
I started to interpret it that if it turns purple, then it's interacting. But if it's yellow, and then I was like, no, that doesn't make sense either. I don't know. Which is why, again, if you just cover up, I'm like, I have the paper in my hands. If you just cover up the images and look at the graphs, even though I don't like the way that they showed statistical significance, they're actually pretty informative by themselves.
Yeah, the
So I
graphs make
think,
complete sense.
yeah, I think that they just added those to because hey, look at us. We spent so much money on our fMRI. One of those machines. I'm gonna take a guess here for let's see. Was last time I looked at them, they were around $100,000 for a mouse for a rodent. So now they may have come down in price, but anyway, sorry. Yeah. So what was the second question?
Uh, how come so C and D, it looks like C is the female analysis and D is the male analysis. That's what it says. That's pretty obvious. But how
Mhm.
come they picked different regions of the brain to analyze for female and for male? Because I mean, like
They didn't.
logically, I would think you would want to analyze the same ones to be able to
They
compare it
didn't.
well.
They didn't pick different ones. The differences are what they're showing. And it
So
just so
then the
happens
question is,
that in the two different in the male brain and female brain, which exist. So I'm sorry, woke people who are in this group. There are significant differences between male and female brains and wiring. Okay. And so what happens is when you see a significant thing pop up, then that's what they're showing. They're not trying to get you to compare the images because they're not that informative. So they're just showing you, look, here's where it was upregulated or different in the female, and here's where it was upregulated or different in the male. Make sense?
That makes sense. That's what I figured it probably was. They were showing the significant regions.
Yeah. But they still have to do this. Like that, I'm being a little critical, but as you'll see in figure 3, for example, like sometimes you got to do the nitty gritty stuff before you can present the the cool findings. Okay.
I was gonna say if Tyler wants to do figure 2. I don't know if I need.
All right. Let me see what I have. Okay, so for A, the bar there on the bottom with the colors, it shows the functionality based on the sex genotype.
Mhm. Yeah, I mean, we really don't know, Tyler, right? That's what we were just talking about. Is like, I can't even interpret that. I don't know what they're trying to show us with the color.
Yeah. For the right graph here,
Yeah.
it shows the connectivity strength of the fMRI is increased in is higher in wild type females than UB3A mutant. So the UB3A2 mutant would decrease the fMRI connectivity strength. Females. And for males, it's close, but it seems to be the other way around. But yeah, they're around the same connectivity strength.
So if I can interrupt, Tyler, because I want to see if you guys remember this, but right now in my honors bio class, we are talking about statistics and how bad biologists are at it. In this case, everyone, right? We're bad at statistics. I want to get this right. What are the things they're actually comparing in A? They're comparing males versus females. They're comparing wild type versus the mutant, and they're seeing if these two things interact with each other to either increase or decrease depending on what the effect is. And that's called an interaction, which is really hard to explain without talking about abusing twins, which is the main example that I use, Maggie and I were talking about it earlier today. But like, an interaction is when something the effect of something gets exaggerated. The effect of a genotype, what your gene is, gets exaggerated because of the environment you're in. And the reason I'm bringing this up is I just don't like the way that they are showing that interaction with the hypo SS asterisk. You see what I mean? Like, to me, it's that what that means is that females are different than males. Are they? Lila, are they in this one?
Are you saying for this one?
Yeah.
I'd say not necessarily. But actually, I mean, like in some of the later ones, I feel like it's saying that it's presenting differently because it's talking about specific like brain features, and it's showing that like men would have like more repetitive behaviors and like cognitive rigidity a little bit, whereas like for women it's more like brain features that have like subtle like emotional regulation issues. That's kind of what I wrote down, but I don't know how
Yeah,
to.
and I think you're right. I think you're right. And I'm looking at what it says in the paper, and it's kind of unclear. there's definitely a statistically significant interaction, but I don't know if there's just a simple main effect from sex or a main effect from the genotype. So anyway, go ahead, Tyler.
Okay. For B, it's fMRI connectivity to prefrontal cortex.
Yep.
The images here show changes in the hypothalamus, thalamus, and prefrontal cortex. It's in red here. Same thing for the other one. Yellow is increased functionality, and blue would be decreased. For the graph, fMRI connectivity strength is about the same, while being more spread out for wild type females and clustered together for UB3A mutants. And for males, the two are wild type connectivity strengths, and UB3A2 mutant strengths are clustered as well.
You want to add anything, Lila?
I don't think so. I don't know if you were expecting me to add something like if there was something I should have said, but I
No,
think
no.
if that's it for
Yeah.
Tyler, then figure 3 is Maddie. Maddie, oh my God, Maggie. I'm so sorry, my queen. I will never do that again.
It's okay, no problem.
Hey,
Okay,
Maggie, don't screw up.
thanks, Dr. Char.
Yeah,
Um,
I'm encouraging.
for figure 3, to my understanding, they tried to like create a mouse for autism so that they can kind of like test the characteristics of autism. Which, as Lila said in the beginning, the general characteristics are the intellectual disability, social disability, and then the repetitive behaviors. So starting with A, they did a rotorod test, which is basically where they put a mouse on top of like a rod that's like continuously moving at the same pace, and they kind of wait till the mouse falls. So going on to graph B, you have the latency to fall, which is basically the time for it to fall. And then on the x-axis you have the like sex of the mice. And then they're testing the wild type and then the UB3A2X. So here it wasn't, so there was a significant difference, but the thing that they're testing is the like interaction significance. So there wasn't much like there wasn't a significant difference. And
What
then
was the
C,
significant difference, though? It was because I didn't like this either one. Which one was significant? It says it in the figure legend, right?
um
It says, yeah.
I think I was gonna say I don't want to cut off anybody. Are you talking about like for all of them, like which one was most, I think they were talking about like self-grooming was the one that was most significant.
No. I
Oh,
meant for B. They
mm-hmm.
make the point that there's, as Maggie just said, there's no interaction happening between genotype and sex, but there were what we call main effects, right? Is there a difference between the sexes?
I think they said the male ones fell off the rotorod faster.
That's pretty interesting, right? Because what's the rotor-rod testing?
isn't it testing the intellect of the mouse?
No, 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 no. It's because it's a motor thing, right?
Right, yeah.
Cam just got excited.
I was gonna say motor skills.
Yeah. And why?
Well,
Doesn't
it's
that
like
seem out of left field?
I guess you were studying parts of the brain that had to do with motor control a little bit before, and you kind of that might align with the third part about like repetitive behaviors and like or like yeah posture and stuff. I don't know. Yeah.
Right. Right. I mean, so this is something, hey, freshman, okay? This is something that it may seem boring at first, but you have to do it, and it is controls, right? You have to run controls. If I, the example, this is a gruesome example, but I'm going to hell with lots and lots of mice playing band instruments. So, if I were to just walk up to a mouse and stick a needle in it through its skull and just go like that with a needle and just like screw up its brain, and then it showed all of the other things that Maggie's going to talk about, that she hasn't yet talked about, but all these other phenotypes, couldn't you just be like, yeah, but you just damaged their brain. Of course they're not going to work, right? And so in this case, you want to test, hey, is this genotype, is the mutant actually throwing everything off or just things that could be specifically linked to autism? So in this case, you put a mouse on a roto rod and see if they can stay on it. This isn't torture or anything. They kind of find it fun, I think. So you put it on, and this rotor rod just continues to spin, and they have to stay on it. And I thought it was pretty cool that actually males fall off sooner than females. Females have better balance. The female mice have better balance than the males. But there seems to be an effect of the mutant because you can see it in the females. David, you're still showing, right? If you could just use your mouse to kind of show the female gray ones, the light gray ones, right, are lower than the dark gray ones, and so are the males. Males are the light ones are lower than the dark ones. So there is a main effect. They're just not interacting. So is that autism? Like this is going to get to the question of, like I kind of put this to Maggie. Maggie, how do you know if a mouse is autistic? Right?
Yeah.
Do you have a conversation with it? Does it not make eye contact? Like what is how do you do that? And that's what they're trying to do. They're trying to establish that look, we have a model and we've gone and tested these other things. So we're going to test the motor skills and see if they're screwed up, if they have screwed up motor skills. And then we'll go look for other phenotypically stereotypically autistic phenotypes. Okay, so I'll shut up now. Go ahead, Maggie.
Okay, so and then for figure C, I thought that it wasn't too informative, so I'm just gonna skip past that one. And for figure D, they're kind of testing the repetitive behaviors of the mice. So mice obviously groom themselves to like wash themselves off. But it's usually quick. So they here are testing the amount of time it took to for themselves to groom. And for graph E, it's again showing the time on the y-axis, and then the x-axis is showing the sex of the mice. And there was a huge spike for the mutant male mice. So that kind of helped to like show their hypothesis in the beginning. Um, and like an example of like repetitive behaviors would be like Asperger's, where they often like continue to they start at one point, and to get to the next point they often count their steps. So it's similar to how the mice would continue to groom themselves, like the male mutant. And then moving on to figure F. Um, so this one they were testing, I think, the like how well the mice could socialize. So starting off with our star, the white mouse. So he is going to be used continuously throughout this. So they first start off by putting the white mouse in with a brand new mouse, which is the gray mouse, and they leave them to kind of interact for a minute. And then they take the white mouse and isolate it for 3 minutes. And then they add a new mouse in for a minute for them to interact again. And here, I believe there was like pretty much no difference between the two. So, yeah, I mean, that one didn't help too much. And then moving on to it was the response to novelty, right?
Mhm, mhm.
So they did, I believe it's called um, I don't remember the name of the test, but the three-chamber test, there we go. So with the three-chamber test, they have the white mouse and they put it into like a cage with like three different chambers, and inside that like cage, they have a smaller cage with another like a new mouse, and it's first testing like how social the mouse will be with that first mouse, and then they as like that the white mouse interacts with the gray mouse, they add a new mouse into a second little cage, and novelty basically means new. So they're testing if the white mouse will go and investigate kind of the other mouse or if it'll stay with its friend. So moving on to J and K. So J is showing the social ability index, and then again the sex of the mouse, and then K is also showing the novelty index, which is also showing the sex of the mice. And again, I don't think it was very significant. There wasn't really a big difference. So I think the only one that like really helped prove their hypothesis was D and E.
I was gonna say I don't want to cut you off
Lila,
because you're. Sorry.
no, go ahead.
I was gonna say you ate that up. I literally couldn't have said it better myself. But I was gonna say, yeah, it almost irritated me every time I'd look at a graph and they'd be like, by the way, this isn't significant. I was like, thanks for letting me know. But yeah, I agree. I think they didn't show anything new about females from this. They only had that men in general, like male mice in general, were falling off and had worse motor coordination, and then the excessive grooming thing. Sorry. Well, why didn't you? I don't know.
Jerry, I am so offended.
All right.
Yeah. Well,
No, that was
yeah,
it.
so I wanted to ask you, I felt like this this is a feeling, hence I said I felt like um this kind of weakened their argument a little bit about having created a mouse model of autism when it's not if you go back to what the David, go back to the triad real quick. Um when okay, fine, we've got repetitive behaviors, which is the lower right corner of the triad, right? Yeah. But the lower left seems unperturbed, and the upper right upper one also seems somewhat unperturbed, at least in these mice. Am I reading that wrong?
But could you reiterate the part right before you asked that? I'm sorry.
They seem it seems like only one part of the triad is represented here, and that's grooming behavior. It's almost like they're saying that the sex by genotype interaction doesn't affect sociality, it doesn't affect novelty responses, or like communication, but it does affect the obsessive compulsive or the compulsions that you would find. Is that the
Yeah,
claim?
well, it's weird because I feel like before I agree that that weakened their argument, but I don't think it completely wrecked it because I feel like figures 1 and 2 were kind of decent in terms of saying, oh, it um there was hyperconnectivity in this part or like reduced connectivity. Like I feel like that was decent, but then I don't know if the way that they tried to measure the behavior just wasn't, well done, and that's why it's not demonstrating. But I if
No,
I just
no.
saw. Hmm?
No, I didn't think it wasn't well done. I just was.
Well, I don't know if it's not that they did it incorrectly, but just that maybe it wasn't the proper way to demonstrate something like that, or maybe it, something like that. I don't know. Because, so wait, Maggie was saying, I'm sorry.
I just said it, I said I think it's a little bit too broad. Like the
You're
test.
saying, yeah,
Maybe.
I could see that.
Yeah. Okay.
We could do figure 4 and 5 if we're done with that one. My only concern is it is like 9 o'clock, so I don't know
Yeah,
how much
time.
you want to dwell
Right.
on those. Or
You
if we,
guys, well, you guys had trouble with 4, you said, right?
are you saying
I
all
think I
this?
got it. I think I got it.
Okay,
Especially if we don't have time, I can
king.
just burn through it real quick. All right,
Okay.
so part A, this is just pretty relatively basic box mixture plotted with that M plus F minus on the left. Those are not gene. No, it is a list of genes. You see that Shank 3 and that other list at the bottom that's in blue text. These are the M plus F minus grouping, and this is where it is an upregulation of females and downregulation of males. That's their production of the gene under different variants. You can see the wild type on the right under female and under male, and then the mutant on the left in both. And then M minus F plus is the same information. It's just, thanks, Dr. Char. It's just it's the same information, but with the M minus F plus, which is going to be where the males are downregulated and the females are upregulated. Sorry, my notes are horrendous. But this is essentially saying as we come down into B, where we're just looking at results with some pretty horrendous abbreviations that male mice are more likely to have this autistic gene under that mutant. So you look at those, it's called a heat map, or it's plotted not logarithmically here, but inverse logarithmically in D. So you see those red highlighted boxes of the DUP15Q. This is just some form of autism. I forget what the formal name of it was, but you can see that's most intense in those male downregulated in the mutant group. Up to C, you're looking at the top of the graph mainly for the biggest point. See that red X, that's the percentage of M minus F plus genes on the X chromosomes, I believe under the mutant. But I don't know if you guys have gotten here in bio yet, but you'll know that X chromosomes or X chromosomal linked diseases are going to descend down the male lineage because there isn't typically a comparable component on the Y chromosome. And
No, no, wait. You're gonna see it more often in males, but it's actually
more
descending.
often. Okay.
It's descending down the
Yeah,
phenotype.
descending maternally, but in the males, yes.
But you see it more often in the males, yeah.
Yes. Okay. So some statistical stuff I wanted to point out. You'll see that bar at 0.15. I believe that is that's indicating a passage of the false discovery rate, which is just some threshold that you need to have for it to kind of indicate that it's not going to be based on chance. And then you'll look at B and D, those boxes outlined in black, which is good for us that are the more significant results. That black outline indicates that they passed FDR. So more significant statistical results than if they weren't outlined in black. And you see again the more extreme values in D, M plus F minus and M minus F plus. Again, that black outline indicating that they are not due to chance, again, are more significant values. Different abbreviations, DHT to start is dihydrotestosterone, which I believe upregulates
It's
which I believe, why is that your enemy?
Because it causes baldness.
Will the mice go bald?
I was gonna say this was my choice.
If they live that long, yeah. The problem is
Wow.
the mouse model they used was actually a strange breed that is not commonly used. I'm not sure why they used that breed, but in some mice, yes. Yeah.
All right. So then after that DHT, that is an estrogen supplement, where you can see kind of darker splotches on that M minus F plus region where the M plus F minus where the females upregulate. that kind of decreases again. And you see kind of the same like inverse reflection of treatment in that dihydrotestosterone. AR and ER targets and everything down from there, I didn't get a whole good grip on. So if Lila, you want to jump in there, that would be great. But if not, no problem. I don't recall it being super significant.
Yeah, I don't think so. I think we should move on to 5.
All right, perfect.
This one's a whole lot of fun.
Yeah, but keep it like super broad. Like
All
just
right.
what is this thing?
All right. So it's super intimidating when you look at it. This is like a visual representation of what it sounds like to listen to Lila talk.
All right, can we kick him off the call? Can we kick him off this call? He did.
I agree.
You didn't review it. No, time.
All right, anyways, quick. It's already 9:04. Um, so this is like a heat map or a map, not really a heat map, a map of the connections between a different genes. So each of these circles is a gene or like the protein that that gene codes for. And then it's divided into 3 groups, green, red, blue. Everyone can see that. And then right next to green, red, and blue, it says what like those types of proteins or genes are related to. So like green is glutamatergic, ion channels, GABAergic, and a synapse. And then red, a lot of these are related to protein synthesis and translation. And then blue is chromatin, transcription related, ERK signaling, and mTOR signaling. And then another important thing to notice, or actually, all these lines between all of them, the lines represent like proteins that interact or genes that interact with each other, the proteins. So ones that are really closely clustered together are, I mean, it's simple. Those are the ones that interact with each other more. And then highlighted in the top right, it says what each of the highlighted ones are. So we'll start with the main guy in this whole study. Orange, kind of in the middle there. That's UB3A. And you can see what it's closely linked to. Um, those those pink ones are sex hormone receptors. Um, oh, I can't remember what the red ones are. Wait, hold on. I think I have it written down. Oh yeah. Those are really important genes for protein synthesis and transcription. I think. And then all these you see all these circles that are highlighted in black or outlined in black. Those are ones that are listed as, or the study says are listed as very closely linked to autism or what did I write? Yeah, linked to autism according to the FARI ASD gene list. Autism risk genes, they call them. And then on the right, B lists a whole bunch of different cell types. So for each of these categories, the green, blue, and then the red. And then to the right of that, it says M plus F minus, and then M minus F plus. So those are the 2 gene sets that are being analyzed in this figure. M plus and F minus is things that are affect the males and not as much the females, and then vice versa for the next one, I think. Maybe correct me if I'm wrong about that, just quickly. Okay, I guess I'm close enough.
No, you're good, yeah.
And then the numbers are what did they call them? They called them um
Yeah, I wouldn't worry about
enrichment
that number.
odds ratio is what they called them.
Yeah.
And
I think if I
sorry.
may, just in the interest of time, I think you already got what was important about this figure, which was an A.
All right.
And I think if you draw attention, David, if you can use your arrow to the middle one that says FMR1, that, for example, is actually the gene that causes Fragile X syndrome, which is one of the more common types of autism. And so all these things are connected to each other because they're all involved in the same, doing the same sort of things to each other. Cool? All right. And then that's good, Cam. Sorry to cut you off. I just wanted to kind of, in the interest of time, get Lila, because you know Lila's going to take 15, 20 minutes on her conclusion.
I'm gonna try my best. I don't know. I'll be like so fast because I know everyone wants to leave like really bad.
The part
Wait,
that
I
you
have to
speed
mention it.
up.
Oh my God. Okay. So, yeah, my overall thing was just trying to say, and now we kind of dunked on it a little bit, so I feel bad drawing this conclusion, but I tried to use iffy language. So I said an overabundance of the UBE3A gene appears to affect brain connectivity, behavior, and gene expression, which were the kind of connectomic, behavioral, and transcriptomic levels we talked about before, um, differently in males and females. So we mentioned that men tend to exhibit more stereotypically like autistic behavior, like we saw that in kind of figures 1 and 2 that they had hyperconnectivity in some in some regions, and we also saw the self-grooming thing in figure 3, and again kind of relating to men, we said that UBE3A gene overdosage could be a factor in the higher prevalence of diagnosed autism in males. I see a bunch of different ratio numbers all over the place. It seems like it's constantly changing, but the constant is that it's almost always more frequently diagnosed in men. So I had a couple key questions which we don't have to answer here, but and I don't really have solid answers to any of them except for the second one where it says if UBE3A overdosage only causes autism in 1 to 2% of the case, so these conclusions applicable to the broad scope of the entire disorder. I would say yes because of figure 5, which Cameron covered, just because the overdosage altered gene networks of that were linked to some stuff where these same pathways were disrupted in other like autism linked genes. So I feel like it kind of says that UBE3A is part of a broader genetic network contributing to autism a little bit. Yeah, but I had other questions like the fourth one I was particularly interested in, but I didn't know what my answer was. But we can go to the next slide unless Dr. Tar, you want to focus on any of this.
Let me interject.
Sure.
So Lila's going to finish the next slide, and I'm going to say, okay, that's the end of the official thing. I'll stick around afterwards. I know Lila will probably stick around if you guys want to then address those where you don't have to be here. If you want to stick around and chat about it, because that's kind of what we do. I know the Journal Club should be around an hour, but sometimes this is pretty cool. Like I thought this was cool. Keep going now.
So for the does this paper belong in science advances part, which is the next slide. So I just looked at what the journal impact factor was, which I believe is just citations over papers published. And I don't have a general idea, like to put in perspective how big that impact is, but I put science and nature and cells, so it seems pretty small in comparison to those. So I kind of mentioned some concerns of like, okay, what do we do with this information if this information is even super relevant? I still think it's relevant enough to be in science advances because that seems like it's not a super duper giant paper. I think if they wanted to be one of those journals, I feel like they'd need, okay, you're making so you made an expression. But I was gonna say like, I feel like to be one of those journals, you might need to have, more clinical relevance, like some way to combat that overdosage or human studies and probably better behavioral results than what they had. But that's what I'd say. If you wanted to be one of the bigger papers, that's probably some stuff you'd probably have to add. But that's all I have. Yeah.
I completely agree. Yeah. Anybody have questions before we end the official part of this? Seeing any hands? Okay, so I'm going to stick around if anybody I'd like to actually talk about the questions that she raised, but I don't want to hold you here. So you can stick around if you want. But good job, Lila, again, and the rest of the crew who took on figures. And let's give credit to Maggie, who is the first person to present from Holy Cross. I thought maybe it would be a junior or sophomore, but it's a freshman. So good job on that one. Yeah, I think this is really interesting. I agree with pretty much everything Lila just said about whether it belongs in this journal. Cool. Okay. So if you need to go, goodbye. We'll stick around otherwise. So an interesting thing about an interesting take home about puberty. Remember, we talked about how uh, people with autism. No, actually, Lila said that when you're young, neurons are forming lots and lots of connections, way more than you need, right? Is that how you put it? Something like that.
Yeah, sorry, I wasn't sure if you're asking exactly. I was gonna say I feel like you have too many synapses
You do,
per
you
se.
do,
Yeah,
yes. Yeah.
yeah.
So you have too many synapses. One might say that this is the reason that children are so creative. Is they have the way I say it is babies and young like infants, toddlers, young children. Their neurons are super promiscuous. They hook up with everybody, and so you have all these connections that you don't necessarily need, and you don't know why they're there. It's just that neurons go through this frenzy of connecting to everything first, and then throughout late childhood, and then yeah, so through late childhood you start to prune these away, except in autism. In autism you have about 16% of the pruning that you see in normal childhood, except when you get to puberty. In puberty you go through this enormous change that I'm sure all of you have had at some point in health class, where they talk about, oh, you're going through a lot of changes and blah. What those health teachers don't really realize is actually it's more true than they're letting on. Your brain is totally rewiring. And so what it'll do is it'll get rid of all the synapses that it doesn't need, including people with autism. Okay? If they make it that far where they're high enough functioning and they get into puberty and all of a sudden the hormones kick in, they will start pruning away all that extraneous stuff that they don't need and strengthen the synapses they do. And on the other side, when you're out of puberty and you're an adult, you're actually far more neurotypical as an adult than you were as a child, to the point where it's like you almost don't know that the person had autism earlier. So that's the silver lining I'm referring to. So if you have a childhood diagnosis of autism and you really do a good job of keeping up your training and challenging yourself, you have to kind of be self-aware that you have autism to do this, and your parents have to be aware of it to give you the therapy you need, and it has to be paid for by insurance, hence the whole thing I was talking about earlier, right? And then get you to the point where your synapses all reorganize to come out with the adult version of your brain, and it's going to look way more neurotypical than it would have had you not had that. So, like statistically, I kind of exaggerate this, but babies and young children have about 2,500 synapses per neuron, and then that increases going through puberty, the creation of the synapses, 10,000 per neuron, and then drops back down to about 2,500 per neuron in adults like me. But those 2,500 actually are meaningful, whereas the 2,500 in childhood were not. If you think about like where, if you hang out with kids long enough, you're like, sometimes you're like, where did they get that idea? Like they'll just say things and you're like, whoa, where did that come from? They have connections that you don't. So they're super hyper creative, and that's good. But they're also going to prune a lot of those away in puberty. And so it's kind of like, what's interesting is why. We actually don't really know. Why is what about puberty and the change in hormonal tone is actually causing that rewiring? And this study is getting kind of at that. I just want to say that again. If UBE3A overdosage can only causes 1 to 2% of cases, are these conclusions applicable to the broad scope of the entire disorder? Does it explain male-female diagnosis ratio?
Because in discussion forums, I kept seeing people that were saying like, oh, like or like in a lot of conclusions people were drawing, they were saying that it explained, oh, like this is why it's 3 to 1 or whatever, is because they're just identifying, you know, men that have it. But then I'm like thinking, okay, well, if men are demonstrating the more stereotypical behaviors, does that mean that only they're getting what we consider to be autism, and should we change those diagnostic criteria so it fits women, or will that change what we consider to be the disorder? Yeah, like I don't know if we should change it to be like fair per se, or if we're gonna start to broaden it too much. That was kind of my thing, and I think all those were kind of like to my last question, but I don't know.
And you came up with, yeah, see, I think that analysis, the way you just put that in a very Lila way, but the way you just put that is like extremely relevant. Like it's a discussion that we've been having in the field for a while that really, and this didn't come up at all during the official part, that really mirrors the discussion we have about ADHD, right? ADHD is also 4-to-1 male to female. Why? And then you're left with like I'm more and more I think that from as a my professional opinion on it is that it's not a disorder, ADHD. ADHD is not a disorder. Instead, it is just a manner of processing the world that's really better suited to a different time when there's like hunter gatherers. Well, can the same be said for autism? Is autism actually a disorder, or is it just an alteration of developmental programming to create a person that has a certain skill set for a certain time so that they can actually thrive? And I mean, certainly you look at somebody with Angelman's. In the olden days, those poor kids are dead, right? They are not going to survive. But if you look at the other end of that autism diagnosis, right, and you get to say Asperger's is those people tend to be very highly intelligent, at least as the way we measure it, which you might argue, if a feminist might argue, actually the way we measure IQ as intelligence is actually built to measure what men are naturally good at and not what females are naturally good at, which is not an irrelevant statement, right? It's I don't really agree with it myself, but I'm not dismissing it outright because who's making the test, right? Maggie?
So I just wanted to ask, like, going back to what you said about like ADHD and then like channeling it like it onto autism. How would you like describe like a disorder then? Like, because you mentioned that like ADHD and autism are kind of like alternate ways of like viewing life. Does that make sense?
Yes, yeah, definitely. Um, and that kind of also touches on the problem that you poor kids have with the glorification of disorders, right? Let me start there, where in social media, everybody broadcasts about their mental health disorders, right? It's like a badge of honor. It makes them special, right? Let me tell you about my depression today. Well, I come from a generation where it's like, you don't talk about that. And there's a reason, because it's not good. There's That's not something to brag about. That's something, and it's not something to be ashamed about. That was like, as a person who actually went around giving talks about mental health disorders and stuff, I constantly said things like, we want to destigmatize these mental health disorders so that we could talk about them and not be embarrassed by them. That in no way did I mean everybody should have one and they're really fun. You should all try it. That's not what I meant. But that ended up being what happened post COVID because when you're locked up in your room, unable to go outside and actually do the normal human thing, you start getting you start getting antsy about like who are you, what's your identity and stuff, and you realize you don't really have one. Right? You're so young, you don't have an identity. And oh man, it takes a lot of work to get an identity. Is there some shortcut? Yeah, I'll just call myself special with this other diagnosis, right? Well, that doesn't work because the word disorder has a meaning. Disorder means that it's actually disruptive to life. And notice it's being used now for the exact opposite purpose, not to disrupt, right? Um, but to instead draw attention to a positive thing to you. Attention seeking, very narcissistic, And so that I know people, kids. I know I've had kids that I've worked with who have significant ADHD diagnoses that are where it's really, really disrupting how they live in the modern world. And in that case, it's a disorder, though its origins is what I'm talking about. Like, is it actually a disorder in an environment where we're supposed to be hunter gatherers or whatever? No, I would argue probably not, but it is definitely a problem in our society when it gets to a certain point. But then it's also used for, well, I won't even talk about that. Yeah. So disorder, something that actually negatively impacts you or the people around you, right? Uh, Connor.
I think it's really important to know that autism is a spectrum, and some people are more disabled than others because some people need more support than others, not just Asperger's, but it really is disabling to many people.
What if I told you it's not a spectrum? Think I could convince you of that?
Mm,
So, no. See,
Wait.
so you guys get we have this.
He's gonna gaslight us.
We have. What'd you say?
He's
What'd
gonna
you say, Tyler?
gaslight it. You're gonna gaslight us.
No, I mean, let me see if I can gaslight you guys into
I would say
believing it's not.
I have a thought, but could you before you explain, just like an idea, because I've thought something similar, but I'm thinking of it differently than you are probably. But I want to say it before you say it, just in case it's like I don't think it's the same thing. But I feel like when people talk about the autism spectrum, they're talking about like from like neurotypical or like almost neurotypical to like okay, what do you not like that phrase? So you're making that face.
I
I don't.
don't, and I used it. Didn't I use it like 5 minutes ago? And
Yeah,
when
it's just
it came out of my face, and I almost wanted to punch myself because
I
I
just what am I
hate
supposed to
that
say? Like
phrase.
what am I supposed to say?
No, I think you're right. I'm just being
Yeah.
nitpicky. I don't. That's because it's new. We don't that 5 years ago those words didn't exist.
Yeah, but I'll see people often where it seems like they have like a couple symptoms and they consider themselves on the spectrum then, and they're like, oh, everybody's on the spectrum. And I really don't like that. I feel like the spectrum should be if you have all the symptoms, just in terms of how it affects your functioning. Like I feel like that's what the spectrum should be. But often when I hear people talk about the spectrum, they're talking about it like, oh, I have trouble making eye contact, so I'm on the spectrum, but I'm on the super high functioning end because I'm like I have this one symptom, and it's just like that's how I see it. But I assume you're talking about something else. But like,
No, I'm not. That's exactly what I'm talking about. Yes.
I
Um,
agree.
yeah. I think that you have to, and this is very cynical, but you have to kind of think of why do we even have the definition? What's the purpose of it? Think about that. Can somebody answer that question? What is the purpose of even defining it at all?
Because humans are crazy and they need
We
basic.
like to label things.
Oh, I think you both, that was what you were going to say, right, David?
Yeah, I just said they need to organize things. Yeah.
Right. Yeah. So in our attempts to organize and categorize things, we need to label them.
It's
Sure.
important.
But
Sorry.
oh, I was going to say it's more cynical than that. What were you going to say, Connor? Go ahead.
It's more important to do that because you need to find exactly what the people need, what they need
You're
to.
getting
For
there.
the support.
Keep going. Keep going. You're getting there. Get there. Keep going. What else? Why? Why do I need to do that? For what? Like, think about, well, how many years ago was it? Let's say it was 2000. What year? I want to say it was like 10 years ago. About 10 years ago, the DSM-5 came out where they took Asperger's disease as, or as,
2013,
They
I think.
renamed it.
and they stuck it into the spectrum. And people who had Asperger's, and I have a bunch of friends who have Asperger's, were pissed. Like they were like, I don't have autism. Why are you sticking me in that group? Because what's the point? What's the point of defining it? And this is real cynical, but it has to do with money. If you have Asperger's in the old system, you're functional. Why should the insurance companies pay for you to get any type of therapy to help you with things that you're practicing things you're not good at? Like they are somewhat socially awkward, but they have the repetitive mannerisms and compulsions that you see in autism, and they have the hyperfixation you see in autism, um, and the kind of, let's say, lack of interest in social situations, but they're not actually in any way intellectually disabled, right? Well, those two things, those two parts of the triad are kind of important for human interaction. So wouldn't it be nice for them to get some therapy for it? Nope. Insurance says no. So the American Psychological Association goes, okay, takes it and sticks it into autism artificially, right? Well, then, if they can do that with that, can't you say that pretty much the whole thing that's called a spectrum is just a convenient conglomeration of things that need help? You see what I mean?
Yes.
So, and let's go back to ADHD. If you buy the argument, and I don't even know if I buy this argument, but it's fun to make the argument. If you buy the argument that in a hunter-gatherer society, it's actually a worthwhile thing that is selected for, right? ADHD. If the zombie apocalypse happens today, I'm going and getting somebody with ADHD to join my team because they're pretty good at picking out novelty really quickly, right? They notice things very quickly because their attention's so bad, right? Um, and a colorblind person because they can see movement better. But those things aren't really, they don't need to be together, or even they may not even be the same thing. But the outcome of whatever it is that's causing it seems to be similar, so we artificially group it into what we call a spectrum. And the thing is that if you extend the spectrum, it includes all humans. But what's the value of it, right? So there is value in saying it's a spectrum, even though that categorization may be somewhat forced. Do you see? At least that would be my argument. Maggie, in the post talk, we usually just don't. We just talk.
Okay, cool. So I just wanted to ask for like OCD and whatnot. Like, is that tied in? Like, I don't want to say like under autism, but because like I remember when we were talking, you mentioned that like one of the common characteristics of autism was like compulsions, and OCD also relies on compulsion. So I was wondering, like, do those two have anything in common or no? I mean, other than that, but
Yes, and the treatments are similar. People with significant, and I think this is actually reduced being, excuse me. I think that this practice has kind of gone by the wayside, hopefully. But one of the treatments for early autism is to give these children antidepressants. The antidepressants will then lower the amount of obsessive compulsive tendencies. Problem is that antidepressants during development really screw up a whole bunch of things, including vision. Are the compulsions something that is causing significant disruption in your life or the life of the people around you?
I wanted
See,
to
Connor.
go back to like the question you asked like a couple minutes ago. Um, I'm not entirely positive if it was like a question meant to be answered, but I kind of just wanted to debate on it. You asked, I think, something along the lines of like, is do compulsions like disrupt your life and like everyone else's around you, I think you said? Um, what's your answer to that?
Any of you guys can jump in. This isn't about me. But it does it. If it does, then yeah. If it's the type of compulsion that does, biting your nails, does that affect anyone around you?
No.
Not
I mean,
really. I mean,
oh,
you
all
can
right.
think of
Isn't that
cases.
subjective at times?
Yeah, I was gonna
Yes.
say it, yeah. I feel like when I think of disruptive, I think of disruptive to the person's own lifestyle because that's just easier to measure. Because like I could think of like for some reason I tweak out whenever somebody like makes any repetitive sound or any repetitive movement, like if someone's bouncing the leg, even if it's been for like 2 seconds, like I just like I tweak out. But like yeah, that's disruptive to me, but it's not disruptive to most people. Like nobody else in the room cares that someone's bouncing their leg or twirling their pen or something. So I again like and even though like you could say, well, you're an anomaly and like the general public doesn't see that as a disruption and therefore it's not a disruption. Like I just feel like in general that measurement is like weird. I'd prefer it as just is it disruptive to you? Because in that case you'd want treatment, not because it's disruptive to everyone else. Like I remember my brother, like his first grade teacher wanted him to get a diagnosis of ADHD and get put on meds because it bothered her a lot, but it didn't actually affect his learning at the time very much. And so I feel like, yeah, I imagine like that is whack. That's all. That's it.
It's like me saying Lila needs to go on meds because she disrupts me.
Leave her alone.
Hey, thank you, Maggie. See, she's
No,
like she's a better friend than anyone else, and I've known her for like 2 days. It's great. It's so good. But also, actually, no, I have something else to say, but I'm gonna wait until we like start.
well,
Keep going.
Did you have something to say in response to my thing or no?
I'm trying to figure out what your thing was because
I could
it was buried in a whole bunch of Lila-isms. So
and anecdotes, lovely anecdotes about my pleasant, beautiful life. Yeah. No, I was gonna say like I what I meant is when people are talking about like is it disruptive? I think it matters more whether it's disruptive to the person like themselves rather than anyone around them. I mean, like yeah, obviously if my disorder causes me to like
gotcha.
hoop and holler all the time
Yeah.
and people are annoyed
Like
by it, like that
Tourette's.
might be some cause. Yeah, but like
Like
yeah, I
Tourette's.
should said Tourette's. I don't. Yeah.
Right. Okay. So the classic example that I use in my teaching, I think that a lot of other neuroscientists use is drug addiction. Right. At what point is something addiction versus dependence? So dependence means that in order for your body to physiologically work correctly, you need the drug. Right. So you're dependent on it, but that's fine. Right. Hold on. Okay, yeah. So that's fine. But if then you actually change your lifestyle in a negative way to get the drug, and it negatively affects you and it negatively affects the people around you, then you're talking about a drug addiction that is bad. Another example is anorexia. So anorexia is not a mental disorder. It's a physiological um, is it a symptom? A syndrome where you are bringing in fewer calories than you need to maintain homeostasis and stay your weight, right? To stay at regular metabolic homeostasis. That's what anorexia is. And it usually means that you are actually in a dangerous um that you're losing weight in a dangerous way. Anorexia nervosa is a psychological condition where you are doing destructive things, destructive behaviors affecting you and the people around you, causing anorexia. Do you see?
I was gonna
So,
ask that because I thought that anorexia was like, um, whenever like, because I remember in health, whenever we were learning about it, um, they were talking about how like, oftentimes like people with anorexia nervosa would like throw up their food after eating it, just because like they almost like wanted to give off the impression that they were eating, but then like their body just would not take it in.
yeah, that's a different type. That's actually called bulimia nervosa, but it caught bulimia causes anorexia. It's the anorexia that's the physiological problem that kills you if
So,
you take it to an extreme.
so it's not the like mental condition of to anorexia that hurts you.
Well, it it certainly makes you it certainly creates the problem, right? Because if you didn't have that mental condition, in the case of anorexia nervosa, if you don't have the mental condition, you're not going to be anorexic. You'll eat, you'll be fine.
How can
So
you
it's kind of upstream of it.
how can you be sorry, I think you just said this a couple minutes ago, but I forgot. How can you be like anorexic without like having the mental condition to it?
Okay, so nowadays in the era of Ozempic, you're going to see less and less of this, but because Ozempic's in the semaglutide is amazing. We should probably try to do a paper about it because it's everybody's talking about it. But 10, 15 years ago, if you had morbid obesity, there were a couple different surgeries. You could do a lap band, which is a band that they put around your stomach to shrink it, or a gastric bypass surgery where they pretty much kind of remove most of your stomach, leaving you just a little bit so that you can't eat very much. And that helps you lose weight because it's hard to eat very much. The problem the problem with it is it caused complications of anorexia in these morbidly obese people. So you'd see a woman, I remember watching this on TLC. This was like 20, 30 years ago. Yeah, it would have been almost 30 years ago, where she's in the hospital. She had gastric bypass surgery a few days before, and now she is losing weight precipitously because she can't get enough calories. And even though she's morbidly obese, you still need readily releasable fuel to keep alive. And so you can't, the human body, especially one that had been sedentary, is not set up to convert fat fast enough into ATP to keep the person alive. And so it becomes dangerous. And so you can have anorexia in a person who is morbidly obese. See what I mean?
I was actually wondering like that, like how long can like a really fat person live? Like off of just like their fat. Not long, I
Yeah,
guess, based on what you're
not
saying.
long. Right. They can't
Because
convert it
what, it just
fast
takes
enough.
a long time to convert that to. And when does that kick in? When you're in a calorie deficit, right? Then they start, or is that happening all the time?
Yeah, so not in them. You actually see it much better done in long distance runners, right?
So even if I'm getting, let's say I burn 2,500 calories a day,
Right.
and I just took in 2,500 calories.
Yeah,
Am I still getting energy from my fat reserves?
not really.
No.
And energy in. So to be healthy, you want to be at an appropriate weight for your frame, and energy in should equal energy out.
Yeah.
So if you're going to use 2,500 calories, you should be eating 2,500 calories.
Mhm.
But what you're seeing is people are eating 5,000 calories using, let's say they start at 3,200 when they're teens. Oh, shoot, Lila, I never talked about puberty.
That's my bad. I was supposed to
Yes.
remind you and I didn't. I'm sorry. There was so
That's
much
okay
going on in my
because
brain because I'm always
we're still
thinking.
on video. Just remind me to say it in a second because we're
Okay.
still recording. But um, you find that as you get fatter, you become more sedentary. So you are not driving the activation of enzymes, the expression of enzymes that will do the job of converting fat into useful stuff. So instead, you're storing it, and as you get fatter, you're becoming less and less active, and then storing more fat. It's a vicious cycle, right? And so if you look at marathon runners, long distance runners, they're super, super thin, and they actually run on fats. They don't do carbo loading the night before a run. If you look at them, you're like, you have zero fat on you. What are you talking about? So they have enough fat and they have all the enzymes ready to convert it very quickly into ATP so that they can just keep running. Whereas if you said, okay, Chark, go and run a marathon. First of all, I would just die right there from the thought of it. But if I went and tried, say 10 years ago when I was healthier, I could go 7 miles. And this is actually true of most people. You could go maybe like 7 miles and be like, well, this is fine. I don't see what the problem is. And then you just hit a wall because I'm no longer I no longer have enough carbohydrates as glycogen to convert it to glucose, to convert it to ATP. And since I haven't trained at all, I don't have a way of turning the fat that I have into usable energy in a in an efficient way so that I can keep running. And so these people they end up having to like either walk the rest of the way or just drop out. That's like training. What's it do? Well, training really is all about did you set up your metabolism to do this strenuous activity?
Sorry, you just saying that like about like wasting energy and whatnot reminded me of like um when you were teaching us about like the Krebs cycle and whatnot. And I because I was studying with my cousin from like in med school, she was showing me like her textbook compared to mine. And I just wanted to yell at you for saying that the Krebs cycle made ATP because it made GTP. But I felt the need to argue that with you.
It also makes GTP. Yeah.
Does it make both?
Yes. Yeah. I just I cut out the GTP because that's just too much information for a high school.
What is the GTP?
GTP. So it's funny. There's also so what is it? What's ATP made of? It's a RNA, right? It's
Yeah.
adenosine, right? Triphosphate. Adenosine is a nucleic acid called ribonucleic acid, RNA. But that's the A in the, right? And what are the other letters for RNA?
What do you mean?
Well, what the code, the the monomers of RNA are adenosine, guanosine, cytosine, and uracil. And each of them makes a triphosphate version, but they're used for other things. So
Got it. Okay.
GTP is right. GTP is one of the main signaling molecules. It's used for energy, just like ATP, but it has a slightly different role that's been set up in metabolism.
Sorry, about the diagnoses stick, when you were saying like that it was either like A, like money related, or B, it was for like not to feel special. You're saying it's in some cases, not in all cases, obviously, but you're saying like it's about feeling unique, I guess, because you don't have much of an identity. I agree with that partially, but I feel like oftentimes it's just that like there's an issue you're struggling with and you're trying to figure out a fix for it, and so you look for a label. Like I know a lot of times, like I'm really into not just personality tests, but like, you know, I'll take quizzes for disorders I'm pretty sure I don't have. Like I read the DSM-5 and I would like look at stuff just because like I was trying to figure out like a box that I fit in, because A, it's easier to explain to people. Like if you have a bunch of these random symptoms and you're like, oh, by the way, I have autism, then all of a sudden it's not a justification, but it just makes a bit more sense. Like I don't know. So like, and that's why like for so long, like I was looking for something like that, is because I'd have quirks that I didn't know how to properly fix, and I was like, okay, if I do the same treatment as like somebody who has this, I don't know. That's my jam.
I think that we do.
But like,
Oh.
I just want sorry, Char. I wanted to say though, I think that like in some cases that could be true to where like some kids like or adults that like have symptoms, like they want to try and like justify those symptoms, so they kind of want to like figure out, okay, well, I want to be normal too, so I want to try and figure out like what I have so that I don't keep just listing off symptoms. But I will say, just from like personal experiences, there's a lot of kids who kind of like don't think that way, and like Char said, like mainly just do it to feel special because like especially like once you like jump into like high school and whatnot, and you kind of went from like in middle school where they taught like, oh my God, you're so special, we can't break your heart. And then you go into high school where like everything is completely different. A lot of kids will like immediately turn to like disabilities just so that like they get like they feel validated almost. So I would say you're right, but also like Char is also partially right there.
Oh, yeah, I was gonna say, like, I see that a lot with ADHD, to the extent that, like, often I don't know whether they have some sort of like neurological issue, or if it's just like, so many of my friends will be like, I have ADHD, and I'm like, you're just a victim of the TikTok generation. Like, I feel like oftentimes they have attention issues and they just chalk it up to ADHD and maybe get put on medication, but it's not like they didn't need to get put on medication. But I don't know. I had that argument with my physics teacher. What does he know? He's a physicist, but like, I don't know. Yeah.
I mean, yeah, I think that like the main issue with like the TikToks going around now about like the disabilities is that now like the people actually with the disabilities no longer get like validated. So,
It's what when you started talking, I was just about to say, and I actually went and wrote it down. We do a disservice to the actual affected community by watering down the pool, right? So if you're using, like Lila, you said, it's sometimes just it's almost shorthand to say you have a diagnosis, even though you don't, right? So that people understand you, which is what you're going for, right?
I
And
mean,
the problem is that people aren't necessarily looking for understanding, right? You're using it in that way, in that kind of shorthand way, for a good reason. And the people we're talking about are using it for attention, which is not, I would argue, is not a good reason. So I don't, yeah.
That kind of relates to the autism. That kind of relates to the autism thing from the slideshow a little bit, like asking if we should change the criteria so it fits women a bit more. because they fall under the radar. But if it's not affecting them as strongly or they're just perceived differently, I don't know if that's something we need to fix per se. It might be something that you want to fix if you feel like it's unfair, but I don't like it. I don't know if they are different symptoms that are getting demonstrated, how much you need to help the women who they couldn't identify it, in the first place. I don't know if that makes any sense.
Yeah.
Mm.
Yeah. Yeah, that's always a problem. I mean, the one thing I didn't say that I probably should have was that most of the studies we do don't use female mice. We just use male mice. And
They
the
don't use
reason
like a
is,
mix.
and the reason is female estrous cycle, which is their version of menstruation, throws off all these physiological markers and measures that we use to understand stuff. But if you do this is really hard and really expensive to do, but if you control for that, a lot of the physiological changes you see in males are actually much more robust in females than they are in males. But we don't know about it because we didn't, like, if you look at my research, I have one paper where I use female mice out of all of them. And in it, we actually did a statistical analysis and found there was a sex-based difference in the robustness of the circadian rhythm, where females have a super robust circadian rhythm and males don't. But here we are using just males because if you don't, they have their estrous cycle is only 5 days, not 28 days. Okay. So every fifth day, if you're watching their, draw it for you. Yeah. So if you're watching their pre-running, where they start a little earlier every day in constant darkness, on the fifth day, it'll jump forward. And then they'll do it again for another 4 days, and then they'll jump forward. And so it's called scalloping down the actogram. You can't, it's really difficult to impossible to analyze that. So we just use male mice where their rhythms are not as robust. So we kind of shoot ourselves in the foot, but at least it's a little easier to analyze.